about the cholesterol biosynthesis, how it is regulated, and what is its role in the process of atherosclerosis. So first is a brief idea about the cholesterol biosynthesis. Every cell in the body can synthesize the cholesterol, and cholesterol is present in the diet also. And as you know, cholesterol is a very important compound required for the synthesis of very many important substances in the body. The cholesterol biosynthesis takes place in those tissues mainly where there is a functioning HMP shunt pathway. So you know the HMP shunt pathway and the cholesterol biosynthesis, they go side by side, because for the cholesterol biosynthesis you need NADPH. NADPH is required. So NADPH is produced in the hexose monophosphate shunt pathway, and so you know they coexist. These two pathways, right? So if you look at the cholesterol biosynthesis, you can see that it starts with the acetyl-CoA. 2 bonds of acetyl-CoA joins in the presence of aceto-acetyl-CoA synthase. CoA is removed, coenzyme A is removed, and what you get is aceto-acetyl-CoA. It is the same step. It is the same step like the ketogenesis. Same step like ketogenesis. And the next step, one is HMP-CoA synthase. Now you have aceto-acetyl-CoA. To this, one more molecule of acetyl-CoA is added, so that you get HMG-CoA, hydroxymethyl glutaril coenzyme A, in presence of the enzyme HMG-CoA synthase. Now up to this much you can say that there is a common pathway for the ketogenesis and also for the cholesterol biosynthesis. Then from now on there is a different enzyme which is known as HMG-CoA reductase. HMG-CoA reductase, which requires 2 NADPH. This is coming from hexose monophosphate shunt pathway, pentose phosphate pathway, and what you get is mevanoate. So this enzyme is different in ketogenesis and cholesterol biosynthesis. Now it is this enzyme where several substances like even cholesterol itself can inhibit this enzyme. Cholesterol, if it is rich in the cells, it could inhibit this enzyme. So cholesterol itself, by a feedback mechanism, regulates its own synthesis, the cholesterol. Now statin group of drugs also act here. So this is a target for several inhibitory reactions, right? So you get mevalonate, then by a series of reactions, mevalonate is 6 carbon compound, because we have used 3 acetyl-CoA molecules. So 1 acetyl-CoA contains 2 carbons, so 3 into 2 6. So it's a 6 carbon compound, and you need 3 ATPs and NADPH, again NADPH, which is supplied by the this HMP shunt pathway. HMP shunt pathway is the major contributor of the NADPH. So that's the significant. That's why I told you that this HMP shunt pathway and the cholesterol biosynthesis, they coexist within the same cell, because that is required, because NADPH is to be used for the cholesterol biosynthesis. Now next, by a series of reaction, it is converted to a 30 carbon compound known as squalene. This is an abbreviated pathway, there are several steps involved which you did not learn that, that is not very important. So squalene is produced, and then you get another compound known as lanosterol. Now, lanosterol, as the name indicates, it has taken the shape of a steroid. This is a steroid. This is a steroid. So slowly it is taking up the shape of a steroid molecule. So that is known as lanosterol. Then you get some carbons are removed and you get a 27 carbon zymosterol. Now from there you get desmosterol, and then ultimately cholesterol is synthesized, which has got 27 carbons. So cholesterol is synthesized from a 2-carbon unit, that is acetyl-CoA, by sequential addition of molecules. Right. So cholesterol is formed in this manner. So this cholesterol, by a feedback mechanism, can inhibit this HMG-CoA reductase. So it is controlling its own, formation. You also know that there are LDL receptors, which we saw in the last class. LDL receptors. Now, these LDL receptors can be either upregulated or downregulated. Now, if the cells contain less cholesterol, then the LDL receptors will be upregulated, and if the cells contain enough cholesterol, then the LDL receptors will be downregulated. So that is another way of controlling the uptake of cholesterol, whereas this is the actual synthesis. So there are many ways of regulating the cholesterol uptake and synthesis. Right. So this is the structure of a cholesterol molecule. This is actually free cholesterol, where you can see an OH here. There is an OH here, so it is a free cholesterol. We have seen free cholesterol. So if you add a fatty acid here, esterified fatty acid, if you esterify it, then you can see it becomes a cholesterol ester. It forms a ester cholesterol. So major share of the cholesterol in the plasma is in the ester form. And HDL does that actually. HDL, high density lipoproteins, they collect cholesterol from various peripheral tissues. Excess of cholesterol that is collected by the HDL, high density lipoproteins. What is collected is free cholesterol. Now this free cholesterol has to be esterified, and that enzyme is LCAT. We have seen in the last class the enzyme lecithin cholesterol acyl transferase. Lecithin cholesterol acyl transferase that will convert free cholesterol to cholesterol ester. What you need is a lecithin which will become lysolecithin. It will become lysolecithin. The enzyme is, so the substrates are lecithin. What is lecithin? It's a phosphatidylcholine. It's a phospholipid, lecithin cephalate. So the lecithin contains fatty acid, and mainly that is this type of fatty acid is PUFA. That is a polyunsaturated fatty acid. So the fatty acid that is transferred from lecithin to the free cholesterol is a PUFA. PUFA means linoleic acid, or arachidonic acid, right? So it's a PUFA, polyunsaturated fatty acid. So an oil which contains more PUFA will help in the esterification and its further transport from peripheral tissues back to the liver, and from the liver the cholesterol is excreted in the bile. So the PUFA actually helps you to reduce the cholesterol level in the body. So PUFA reduces the cholesterol level in the body, in the blood, and thereby helping in the prevention of atherosclerosis, right? Along with HDL, along with LCAT, PUFA helps. So it is good to take the vegetable oils which contains more of PUFA, right? Polyunsaturated fatty acids. The saturated fatty acids they tend to increase the cholesterol level. The saturated fatty acids tend to increase the cholesterol level. So the saturated fatty acids are palmitic acid, stearic acid, and so on. So the saturated ones should be in minimal quantity. Saturated fatty acids are mainly seen in the non-vegetarian food and also in hydrogenated oils like dalda, vanaspati. So their intake should be restricted and more of vegetable oil, more means with limit. More means not large quantity but in limits. PUFA should be preferred. PUFA, and another one is monounsaturated fatty acid, that is MUFA, and omega-3 fatty acids and omega-6 fatty acids. So polyunsaturated fatty acid may be an omega-3 fatty acid or an omega-6. And the ratio between omega-6 to omega-3 should be around 5:1. Should be around 5:1. But our ordinary diet has got a higher ratio. It is almost 40:1. So there is a purpose. There is a purpose of actually modifying the oil. So this ideal ratio between this one omega-6 fatty acid and omega-3 is ideally seen in walnut oil. Walnuts. Walnut oil, soybean oil is also good. So some oils, which has got a proper ratio. Walnut is, what is walnut? But walnut oil is very costly. Very costly. It is a bit difficult to afford. But that is the most ideal one. Soybean oil is good. So it depends on the type of oil. Because as I told you, this lecithin cholesterol acyl transferase uses this PUFA present in lecithin to esterify cholesterol. The whole thing is present in HDL. The whole thing, you can imagine an HDL molecule consisting of, to start with, free cholesterol. It has got this enzyme, LCAT. It has got lecithin and apolipoprotein, that is ApoA1. So, ApoA1 actually tells you how much HDL is present. ApoA1, if you measure ApoA1, it tells you how much HDL is present. So it is ideal to have higher HDL. So ideal to have higher ApoA1. ApoB, it tells you about the LDL. ApoB, if you measure, that tells you about the LDL, and there should be a proper ratio of ApoA1 to ApoB. ApoA1 to ApoB. That means, or it may be reversed, that is ApoB to ApoA1. That is equivalent to LDL upon HDL. There should be enough HDL present so that the reverse cholesterol transport can be done. Reverse cholesterol transport. What is reverse cholesterol transport? It is from the peripheral tissue back to the liver. Right? So that is done by high density lipoprotein. So it is better to have high density lipoprotein so that your cardiovascular diseases can be prevented. Right? So this was a talking point of that. Now we also saw about the cholesterol. You know that cholesterol is a very important compound. It is highly required. Bile salts, steroid hormones, vitamin D, cell membrane, all these are requiring cholesterol. So enough. But if it is in excess, then the problem arises. Okay, now if you look at this cardio CSD, risk and lipid profile. Now, to know about the CSD risk, you have to measure some compounds in the blood or plasma, that is known as the lipid profile. lipid profile. You can see here, there is a low risk, means as good as no risk. As good as no risk, then you have a borderline risk and you have a very high risk, right? Now, total cholesterol, you know that the normal value is around 150 to 200 milligram. So, ideally your cholesterol, total cholesterol level should be less than 200 milligram per deciliter. So, you have least risk of developing cardiovascular diseases. And borderline risk is known as the 200 to 240. 200 milligrams to 240. If anybody has, there is a chance of borderline risk. And then high risk is that those patients who have a cholesterol level of more than 240, that is the total cholesterol. Now, total cholesterol is made up of various other sub-fractions like LDL cholesterol, VLDL cholesterol, IDL cholesterol. The various types of lipoproteins, the way we have studied. Now, LDL, you know that it is the lipoprotein which carries the cholesterol from the liver to the peripheral tissue. So this is said to be very crucial in its role because they should be kept in check, that is less than 130 milligrams. So the LDL level should be below 130 milligrams, that is a low risk, and you have the borderline risk which is up to 160 milligrams, 160. At any cost it should not cross 160. More than 160 is high risk. So LDL has to be kept in check. There are some ways as to how to keep this LDL in check. I'll talk to you about that later. Now this HDL is the good cholesterol. This is a bad cholesterol. LDL, lethally dangerous cholesterol. Lethally dangerous is not correct. Low density lipoprotein cholesterol is the correct terminology. But to drive the point, you may remember like that. And HDL is the good cholesterol. It should be more than 60 milligram. If it is more than 60 milligram, you can be rest assured that you are protected against the various CVDs, cardiovascular diseases. So ideal to have more than 60 milligram, and 35 to 60 is there is a borderline. Borderline means if the other compounding factors are present. If suppose a patient is having about 45 HDL, 45 HDL, but if his LDL is about 230, if his LDL is about 230, or his BP is more, blood pressure is more, or if he is obese, the waist circumference is more than 80 centimeters in males, 80 centimeters in females, and 90 centimeters in males. The waist circumference, if it is more than 80 in females and more than 90 in male, then that is a risk. That's a risk, waist circumference. You can also find out the BMI, body mass index. But better indicator is waist circumference. How much is it? In females, it should be below 80 centimeters, and in males, it should be below 90 centimeters. Because it depends, there is a difference in the distribution of fat in males and females. In males, it is more deposited near the waistline, whereas it is not so in females. It is in the lower body part, more deposition. So that is less risky. Which one? The female adiposity is less risky because the lower body obesity adipose tissue, it is less metabolic. It is, the activity of the metabolism is less in females, so it is an advantage. Otherwise, in males, it is more on the abdominal region, so it can be metabolized faster, and then it will go to the liver, and in the liver, it will be converted to VLDL. It will be converted to VLDL, and VLDL, it can be converted to IDL, LDL, so LDL will increase. Whereas in females, it is not going to the liver. In female adipose triglycerides, they are mobilized, less mobilized, one thing. Even if it is mobilized, it does not go to the liver, it will go to the other peripheral tissues like muscles, adipose tissue, etc., where VLDL formation does not take place. Where there is no VLDL, VLDL is formed where? In the liver. In the liver. So the fatty acids entering the liver causes more harm, which might happen in the males. So this is a sort of advantage, and not only that, this HDL in females is higher. Females have a higher HDL level. So it is also advantageous. So the higher HDL seen in females is advantageous. Till menopause, this pattern continues. Females have higher HDL, but then after that, menopause, it is the same. The risk is the same. So HDL, maybe HDL is the only substance which is higher in females. Remaining other parameters is more in males. So it affords protection against heart disease. So this is a highly desirable lipoprotein. HDL means highly desirable lipoprotein, high density lipoprotein. So triglycerides. Triglycerides, although not directly involved, you should have a triglyceride less than 200. Each person should have a triglyceride level less than 200 milligram per deciliter, and 200 to 400 carries a borderline risk, and more than 400 it carries a high risk, because elevated level of triglycerides, it can cause insulin resistance. Elevated level of triglycerides can cause insulin resistance. What is insulin resistance? Insulin resistance means, insulin is not able to do its function properly. So if insulin is not functioning properly, although it is secreted, type 2 diabetes mellitus. Insulin secretion is sufficient, but due to some reason, maybe lack of insulin receptors, it is not able to carry out its function. So, high triglyceride level might cause diabetes mellitus. Type 2 diabetes mellitus. So this is about the lipid profile, what should be the ideal and what carries the risk. Now, total cholesterol 200 somebody is having, but if he has higher LDL or lower HDL, 200 is normal, up to 200. But suppose that patient has got higher LDL, more than 170, and lower HDL, maybe about 35, and he has a BP, high blood pressure. Family history is there. Family history is also important. Obesity is there. Abdominal obesity. So even 200 may be higher. 200 although looks to be normal, but that also can be higher if other factors are more. So you have to have a full look, not individually. I can't say that my LDL is only 90, so I will not suffer any attack. It's not possible. It may happen because the other factors also should be supporting. So family history also is very important, right? So you have some idea now about the lipid profile and what are the ways of now other. If you risk factors again coming back now, one risk factor, as you know, is cigarette smoking. Now cigarette smoking, cigarette contains nicotine. So nicotine can bring about vasoconstriction. It can bring about vasoconstriction, and also it may add to platelet aggregation. So cigarette smoking has a bad effect. Not only here, but in many other ways. It may cause cancer also. Cigarette smoking can cause lung cancer. In addition to that, it may affect the heart also. Number two, high total cholesterol. That we have seen already. High total cholesterol, that means more than any value more than 200 is bad. High LDL cholesterol. LDL cholesterol, if it is more than 130 to 160, and if it is more than 160, one should try to bring it back by modifying your diet. If you modify your diet, take less cholesterol in the diet. Vegetarian diet actually does not contain much cholesterol. So a patient having a high cholesterol, if he or she takes eggs, then that is not advisable. Because egg yolk, yellow of egg, they contain large amounts of cholesterol. So eggs will have to be avoided. Non-vegetarian food, which is known as red meat, also is containing more cholesterol. But fish is not harmful because it contains more of omega-3 fatty acid. Fish contains more of omega-3 fatty acid, and mostly it is PUFA, all are PUFA. Saturated fat is less. So fish, Eskimos, Eskimos, those who live in the polar region, they do not get any heart-related problems because their main diet is fish. So that's okay. So the saturated fat should be reduced. You know what is saturated fat? Non-vegetarian fat, excluding fish, is saturated fat. So that should be avoided. Dalda is also saturated fat. It is a hydrogenated fat. So these polyunsaturated fats, they get hydrogenated. So although the taste may be improving because of the hydrogenation, it improves the taste, palatability, but it may cause harm. So that should be prevented. So one way to reduce the LDL is to take a vegetable oils, and everything should be in limit. The limit is the key word. Limit is the key word, right? So, and of course, doing exercise, doing regular exercise, like not you people, you are already active, but if you cross an age of 30 or 35, then you start to put on weight. So you need a lot means, at least 45 minutes every day. Some sort of exercise will help you. Brisk walking. Brisk walking is a good way of exercise. So that will, brisk walking or any, heavy exercise also is good, but everybody cannot do that, heavy exercise.